All right, great. Anything else? Super. Ryan, kick us off, please.
Okay, so I'm going to be updating you on the work that I've been doing for the past 4 months since my last lab meeting. I forgot to change the title slide, so I'm going to skip it. And I'm going to go right into the introduction of T-cell biology because that's the system I'm working in. So the idea is here you have you start with a naive T-cell, which is a T-cell that has never been exposed to its antigen before. And when it, during the course of a normal infection, that T-cell will encounter its antigen, and through a complicated series of signaling mechanisms, it will be induced into proliferating and differentiating into what's called an effector cell. And in addition, it will proliferate very quickly and form a large number of effector cells, which then participate in fighting the infection. And once the infection has been fought off, most of those cells die, and a select few remain as memory cells. And then the next time that same antigen is encountered, so the next time the same pathogen infects the organism, the memory cells can proliferate much more and much more quickly to give a rapid response to that previously seen antigen. So obviously there's a difference between a naive T-cell and a memory cell, and we know sort of some of the mechanisms of that difference, but we don't know all of that, and so that's an area of active research, and that's sort of what my project is intended to get at. Oops, not there. Here. Now in my project, I'm looking at a time course of naive and memory CD4 T cells after activation. And the specific time points I'm looking at are day 0, day 1, day 5, and day 14. And to put those in context, day 0 is obviously prior to the antigen exposure or activation. Day 1 happens during the sort of early antigen recognition and the very beginnings of cell proliferation. Day 5 comes right around the peak of the infection up here. And day 14 is generally after the infection has been cleared and the cells are beginning to die off and become memory cells. So that's the rationale behind the choice of those 4 time points. So the overall sort of abstract goal of this experiment is to investigate the role of well-known histone marks in the processes of T-cell activation and differentiation into memory cells. So to do that, we started with 4 human donors from which naive and memory CD4 T cells were collected. And we activate those cells and we collect samples at the 4 time points that I mentioned. And so that gives us 32 samples because it's 4 donors, 2 cell types, 4 time points. And on each of those samples, we do RNA-seq to get gene expression, and we do ChIP-seq using antibodies against 3 different histone marks: H3K4 dimethyl, H3K4 trimethyl, and H3K27 trimethyl. And then we also sequence the input DNA as a control for the ChIP-seq. So altogether, that adds up to 160 sequencing libraries. So it's quite a large project.
I have a naive question that
Sure.
I've never asked before. So clearly, when you isolate the memory cells from humans, you don't know what antigen activated them before.
Yeah.
But presumably, they all can be activated in that second step using some generic antigen. Is that right?
Yeah, there's some, I forget the exact mechanism, but it's some trick that cross-links the T-cell receptors in a way that is not specific to the actual receptor part. So it unconditionally activates every T-cell. I don't remember the exact mechanism, but it's a pretty well established in the immunology community.
And there's no reason to think that different memory T cells that were activated by different antigens would have any different biology. It's just the MHC that is different.
Well, the T cell receptor that's different.
Yeah. The T cell receptor.
The MHC is the other end of that
right.
interaction.
That's the only thing that's the sequence of that TCR in terms of the state of the memory cell between.
Well, the state of the genome sequence. The only difference is that T cell receptor. But obviously, the question here is what's the epigenetic difference in terms of the histone marks.
And I'm guessing I'm asking between the memory cells that have been activated by different antigens. There's no reason to think that those would be different.
That's an interesting question, but generally they're treated as equivalent. But I suppose it's plausible that if you had different infections that took different amounts of time and effort to fight off, you could end up with a different epigenetic state for memory T cells that are aimed at different antigens. And I guess that's something that you would get at with single cell seq. But this of course is bulk RNA seq and bulk ChIP-seq because it was done, 5 years ago or something. So the original analysis already has a couple of publications. This is sort of the main one. And my current analysis is trying to go beyond this and get into more details. I have a reproducible workflow on GitHub. This is roughly all of the steps in it. And this is the URL where you can find it. So what I presented at the previous lab meeting back in, I think, June, give or take, or July, I forget, I don't know, was the idea of analyzing the neighborhood of each promoter by tiling windows across that promoter. So what I did there was for each gene, I looked at the expression of all the different isoforms grouped by which transcription start site they used, and I picked the transcription start site where the total of all the isoforms for that TSS was the highest. And I called that the representative TSS for that gene in my data set. And then I define that as the 0 coordinate, and I put one 500 base pair window centered directly on that, and then I put 10 windows upstream of that and 10 windows downstream. And then I count the number of ChIP-seq reads in each of those 21 windows for that promoter. And then I do the same for every other promoter in the genome. So now I have 21 windows times however many tens of thousands of promoters, times 32 samples times 3 histone marks. So then I can look not just at the total coverage of the total amount of histone mark ChIP-seq in that promoter, but I can look at where the coverage falls in that promoter, upstream, downstream, or right at the TSS. And then hopefully I can relate that to gene expression in some meaningful way. So this was the one plot that I showed previously, where on the x-axis we have the position relative to the TSS. So this is those 21 windows, each 500 base pairs long. So we're going roughly from 5 kb upstream to 5 kb downstream. And I did k-means clustering of the data after centering it at 0. So I'm subtracting out absolute differences in promoter coverage and only looking at relative differences across the windows. And we have we got some interesting clusters. And then when I looked at I should mention this is only looking at the naive day 0 data averaged across all 4 donors. So a very sort of preliminary simplified way of looking at just a subset of the data just to see if there's some interesting patterns there. So there's definitely some interesting patterns of coverage in terms of the promoter. For example, you got downstream coverage here in cluster 6, you have upstream coverage in cluster 1, and so on. And then the interesting thing was when I took those same clusters, which were determined by the ChIP-seq coverage for H3K27 trimethyl, and I plotted the distribution of RNA-seq expression values, I saw that 2 of the clusters in particular, 1 and 3, looked different from the rest. They had a higher than the expression than the other 4. So I thought, yeah, this looks like there's something interesting going on. It looks worth investigating.
Still day 0 though.
This is still all day 0. So this is sort of a preliminary just to see if there's something interesting worth investigating. And what I'm getting to next is this analysis is kind of limited. This is all done with the day 0 ChIP-seq and the day 0 RNA-seq. So it's just resting naive T cells. And it's not obvious how to easily extend this to multiple time points and multiple cell types. Like I could just make this plot, make these plots for every cell point and time point, but I'm not really doing anything with the change over time in that case. So the other problem is that I'm kind of looking at all promoters with the histone mark present, which means that a lot of these genes, maybe even most of them, aren't even expressed in this data set. So why do we even care about them if there's no gene expression? So I came up with an alternative strategy, which is basically to look at the RNA-seq first and try to cluster based on the pattern of RNA expression across all samples, all time points, all cell types. And then, once I had some sort of grouping based on the RNA-seq, go back to the ChIP-seq and see if there's anything interesting going on there. Just kind of taking the reverse approach to what I did before. And the basic question we want to see is, does the pre-activation promoter state predict the post-activation expression response? In other words, can we tell how a gene is going to respond to activation based on the initial histone mark state? So the first thing I started with was a clustering analysis based on the RNA-seq data. So I basically just took all the RNA-seq data after a large amount of, all of the usual pre-processing and some necessary batch correction, and I ran k-means clustering on it with k equals 5 just to see what would happen. And what we basically see is that there's a central cluster, which is the sort of orange one, cluster 2 in the center, and then there's sort of 4 clusters on PCs 1 and 2. And when we go and look at a heat map of the average expression level of the genes in each of these clusters, we can see that the central cluster 2 has little to nothing going on. And then each of the other 4 clusters, we can kind of categorize them as, well, I should go over what everything is here. So the left half is naive. The right half is memory. Each column is a sample. Each block of 4 is the 4 donors. And we have, like here is day 0, day 1, day 5, and day 14. And then it's the same 4 time points for memory. And then each group of 4 is the 4 donors for that condition. So for example, cluster 1, we can say starts low at day 0, and then it goes up at day 1 and back down at day 14. And similarly, the other 3 clusters down here, they're all up in 2 time points and down in the other 2 time points. So that is the sort of activation response. However, cluster 4 is sort of the odd one out here because we can see that it clearly has a different behavior in naive versus memory. Whereas the other 3 clusters, not counting the one that does nothing, they all seem to have very similar behavior in naive and memory. So we see the activation response, but we don't see a difference between naive and memory, except in this cluster 4. Going onward, I decided to see if there was a difference in the day 0 histone state for these. And, well, there kind of isn't. Well, there really isn't any visible difference. This is the distribution of histone coverages at each of those 21 windows for all the genes in each of those 5 clusters. And you can see there's pretty clearly no difference between any of the clusters. And this is for H3K4 trimethyl at naive day 0, but I plotted it for every combination of histone mark, cell type, and time point, and it's basically the same story, that they all kind of look the same. So the next thing I tried was, instead of taking all genes and clustering them, I said, what if we only pick the genes that have a significant differential expression between naive and memory at least 1 time point? So you can see the results of that here. Again, you can kind of see a lot of the same, where there's a bunch of clusters that are down at 2 time points and up at 2 other time points. Here, cluster 2 is that one that's sort of different, that's higher in memory at the first 2 time points. This cluster looks like some sort of hidden batch effect. This looks like garbage. But other than that, it's kind of the same 5 clusters and then 1 artifact cluster. And again, when we go to the histone marks for each of those clusters, they kind of just look all the same, except for... Actually, I'm not sure why I guess cluster 4 had very few samples in it, which is why it looks funny. But other than that, they basically all look the same again. So again, I wasn't finding any differences in the promoter coverage.
What's in
So that would be genes that were not differentially expressed, so they were excluded from the clustering. But it's still useful to plot them here to see if the differential expression are different from the non-differentially expressed, in terms of, and they're not. Yeah, it looks like they're all basically sort of the same. So the other thing that we can look at is in each of those 6 clusters, we can plot each individual gene's expression trajectory over time as a line. So this, for example, cluster 1 was the one where it's low in the first 2 time points and higher in the second 2 time points. And yeah, you can kind of see that, but you can see it's very highly variable within that sort of paradigm. And it's kind of the same story for all of them, that these clusters aren't really that cohesive and that well separated. And that kind of matches with what we see in the PCA plot before. So the overall conclusion is that I don't really think the clustering approach is working very well for me for this data. The clusters aren't actually well separated on the PCA. They're just kind of different lobes of the same blob on the PCA. And I don't see any difference between them in terms of promoter histone behavior.
What if you took just the most extreme examples instead of like the entire cluster? To the edges of the...
So you want me to basically... What do you want? How would I do that? I'm doing k-means clustering, so that's not a trivial... I guess I could switch to some other...
Oh, yeah, I don't know.
I think in the
I could select for... I could select for...
The highest PCA scores, or the furthest apart in principal component space.
Yeah, that's one possibility. I suppose I could also select for higher fold changes in the differential expression filtering.
Yeah, because in the trajectory it's clearly like a bunch of those genes all go in the middle, but then there are extremes, right?
Mm-hmm.
Mm-hmm.
You could pull those out just based on like, is there like fold change?
Mm-hmm.
Well, I mean, yeah, it's just
Yeah. But anyway, I decided to put the clustering approach aside for now and start with something different. So instead of trying to cluster it and then try to see biological significance in the clusters after the fact, I decided, let me define what I think to be interesting expression patterns and then figure out which genes fit into those patterns. So the sort of decision process I went to for how to classify a gene's expression pattern is at what time point is the first significant change, and in which direction is that change, and then does that gene go back in the other direction? In other words, is that change maintained through day 14, or does it change back in the other direction and revert that change at some point? And I can ask that question for the naive time course, and I can ask it for the memory time course for each gene. And then I can look at genes where the answer is different for naive versus memory and see if there's anything interesting there. And then once I've identified interesting subsets of those, then I can go and look at how the histone marks are behaving in the promoters of those genes. So that I've just started on that, and over here I have a spreadsheet where I've classified, so the total number of genes after filtering out unexpressed genes is 16.7 thousand. How do you zoom in? Oh, here we go. There we go. So rows are how the gene was categorized in naive cells. So for example, day 1 up transient means that the gene was significantly upregulated at day 1, and that and then at some later time point it went back down by a significant amount. So I called that a transient activation. Whereas day 1 up maintained would mean it went up at day 1, and then it never had a significant negative change at any time point after that.
Negative relative to the
previous time point, yeah. To any previous time point. So it went up at day 1, and then all the possible comparisons after day 1. So there'll be day 1 to day 5, day 1 to day 14, and day 5 to day 14. None of those were significantly negative. So what we can see here, and I'd like to thank Andrew for highlighting some of these. The ones highlighted in red are ones that changed in the same that went up initially in both naive and memory, but they went up earlier in memory than they did in naive. So for example, these 4 boxes here, in naive cells they were upregulated at day 5, and in memory cells they were upregulated at day 1. And so the reason these red ones would be interesting is because theoretically these genes would be the ones that explain the stronger and more rapid response of memory cells to an infection compared to naive cells. So a lot of these have very few genes except for this one here, which is the transient upregulation day 5 in naive and day 1 in memory. So that's definitely a subset that I want to hone in on. These ones over here might be interesting as well. There's one artifact I should mention, which is there's no such thing as day 14 transient, because day 14 is the last time point. So if it changes at day 14, there's no chance to reverse that change later. So the red ones are the ones that change earlier in memory than in naive. So those are interesting, potentially interesting for follow-up. The green ones are the ones that go down earlier in memory than naive. So it's the same as the red, but for initial down regulation. As we can see, down regulation is more rare in general, which is not unexpected for T-cell activation. Obviously, T-cell activation is a lot more about turning genes on than turning them off.
Wait, so what is down in day 1? What is that relative to again?
To day 0.
Oh, okay. I'm sorry.
Yeah, day 0. Sorry, day 0 is pre-activation.
Okay.
And then the 3 post-activation time points are 1 day after activation, 5 days, and 14 days after activation. So the red ones are initial upregulation earlier in memory. Green ones are initial downregulation earlier in memory. And then these yellow ones are other. So these over here, for example, these went up in both naive and memory, but they did so earlier in naive cells than in memory cells. So that's kind of an unexpected pattern. And Andrew and I were talking about what could possibly explain that, and the one thing I came up with was maybe a process called somatic hypermutation, which is something that I think happens more in naive cells than in memory cells. And what somatic hypermutation does is it takes the initial pretty good T-cell receptor match to the antigen, and it starts randomly mutating it and selecting for even better matches. So it's optimizing the ability of the T-cell receptor to specifically bind the antigen. And I actually have to check this, but I believe that happens more in naive cells because there's that initial need to optimize it, whereas in memory cells it's mostly already there, and it just needs to optimize it a little further. So it might be a lower priority, something that happens later in the response. But other than that, I don't know. I haven't had. I just got these results pretty much today. I haven't had a chance to drill down further and do gene set testing or look at the exact expression patterns of these just yet. So for now, this is where my presentation ends. I'm looking forward to drilling down on these potentially interesting subsets later.
And so the plan on how to drill down on those is?
Yeah. So step one is that I want to go back and make a plot like this for those specific subsets, just to make sure that I'm actually capturing the expression patterns that I think I am, based on this criteria that I described. And then step two is to go in and plot the, make a plot like this for those specific subsets, looking at whether there's any special pattern in the promoter in terms of what's going on with the histone marks there that could explain it.
And specifically looking for differences in day 0 between naive and memory, right? The question is whether some epigenetic mark in memory is poising.
Yeah, so in the case of the red ones where they go up earlier in memory, I would look at the day 0 naive histone mark in the promoters. I would look at the same in day 0 memory, and I would try to find some sort of difference that represents the readiness of that gene to be activated more quickly.
When you populated this spreadsheet, did you like, how did you sort of do it? Like, did you just manually like run through the not manually, but did you just run through the genes and then see which ones and check the fold change according to the criteria you picked?
What do you mean?
I mean, like when you populated these numbers, right? So did you just go in and just say, okay, these are the questions I had in my end, and so you just picked ones that were upregulated at one point and then changed over time? It's I mean, like, so when you're clustering, what if you cluster based on the fold change over time? So you have.
So that was the clustering analysis that I described before. This is all.
That wasn't the fold change, right? So you were looking at.
Well, it's based on across all 32 samples.
What if you transform that into you pick like the 2 time points, every like a pair of time points, and then you actually transform that into fold change. So for every gene, you would have like the fold change between 2 time points, and then you do.
Well, there's only 4 degrees of freedom. Well, 8 degrees of freedom because there's 2 cell types, right? It would just be the average expression of each time point. It would be equivalent to that. And I mean, if I wanted to sort of deprioritize donor variability, then I could average all 4 donors in each time point. Or what I would actually do is fit a linear model with an intercept for each condition and just take the linear model coefficients. But I don't think it would change much. But what I did to generate this was I used lemma to do a linear model fit, and I tested each pair of time points for differential expression using a log fold change threshold of it had to be either 1.2 fold up or 1.2 fold down. And so then I got a table of, so between 4 time points there's 6 possible pairs, right? So 6 possible contrasts. So I just got the FDR values for each of those 6 contrasts, and then the 6 for naive and the 6 for memory. And I did a global FDR adjustment. And then I just picked the ones that were either significant, and then I got the sign of the fold change as well for up or down. And I wrote up the code to generate this plot. Or I wrote out the code to categorize each gene, and then I use the table function in R to generate this.
How many genes are there? Oh, okay. I didn't see that last column.
Well, yeah, I scrolled off the screen. But yeah, I added the... oh, in case it wasn't clear, static means that it's not changing at any time point.
I was wondering where 10,000 genes went. So.
Yeah. Yeah, there's so static means that it doesn't necessarily mean that it means there's no differential expression at any time point. This column is for memory, and then the row would be for naive.
Sorry, can you go back to that?
Sure.
So let's see.
So the ones like here, for example, would all be genes that did something in naive but didn't do anything in memory.
Right.
And then similarly, the ones here would be things where something happened in memory but nothing happened in naive cells.
And as I was thinking, I mean that's a really interesting group too, right?
Yeah. And and is it a larger group in naive or memory? It looks
I
like...
was just looking at, if you look in the lower right, because it is...
I believe it's a larger group in naive, because these are the ones that are changing in naive but not changing in memory.
Yeah, but no, that's actually bigger, because it's 13,000 minus 10,000, right?
Yeah.
13.5.
Actually, it's pretty close, because it's this number versus that number.
Right, but that one is bigger.
Well, it's 13.5K versus 12.5K.
Okay.
It's not a huge difference. I wouldn't generally consider that a substantial difference. Now, there are definitely differences in terms of what time point those happen at. So yeah, those are another subset that I need to look at, is the one that change in either naive or memory, but not both. But anyway, so that brings it up to basically what I've been doing this week. So that's pretty much the end of my presentation. Unless anyone has any other questions or comments. I'm always looking for, if you have any ideas on what I should do to analyze this data, I'm very open to suggestions.
Have you looked? I assume you're going to do like GSEA.
Yeah.
On this. Are you going to look at transcription factors?
What about transcription factors?
Like maybe
like consider
the
transcription.
targets of transcription factors, is that part of GSEA?
Yeah, that's part of MSegDB. So I can do that. So I could figure out if any of these signatures were driven by specific transcription factors in theory.
I'm wondering, so why do you think that these differences are caused by epigenetics and not driven by transcription factor activation?
So these specific histone marks are known to be maybe not necessarily causally associated, but strongly correlated with gene expression. H3K4 methylation is positively correlated with gene expression, and H3K27 methylation is negatively correlated with gene expression. And they're pretty strong associations. So it might not be a direct causal thing, but it still can be potentially explanatory. And it could be that like histone methylation is what targets a transcription factor to a specific gene. Or vice versa. It could be the transcription factor binds and recruits a histone methylase that methylates the histones.
Cool. Any other thoughts or questions? Great. Thanks, Ryan.